Hello students, in this session we are going to cover an important bacteria that is Vibrio Cholerae, which causes a very important watery diarrhea disease that is called as cholera. You might have heard of cholera, which is a waterborne disease which leads to severe watery diarrhea leading to dehydration and other complications. So this group, it's a gram-negative bacteria, gram-negative bacilli, which contain various species, of which the most important one is Vibrio Cholerae. So it's a gram-negative bacilli. Like the other gram-negative bacilli, you can recollect E. coli, Klebsiella, Pseudomonas, Salmonella. And this is a strongly aerobic bacteria. This can live only in the presence of oxygen. And the very important point regarding Vibrio is they have got a single polar flagella. See, this is the bacilli. It has got a single polar flagella like this. So you can imagine how it will move. It will move very fast. It's actively motile. And you call that motility as darting motility because it will be moving in a super fast way and you cannot trace the bacteria. It's very difficult to trace it in microscope when you take a wet mount and examine. So it is actively motile and the motility is called darting motility. Why does Vibrio shows darting motility? Because it has got a single polar flagella. And the other one, that is, since it is strongly aerobic, it is oxidase positive. It has got oxidase enzyme, that is very important. It is oxidase positive. And the growth is stimulated by the presence of salt. If you provide alkaline pH, it is very good for growth of Vibrio. It needs an alkaline pH for growth. And if you provide salt, it's very good for the growth of Vibrio. So it's salt loving. So how we will classify Vibrio? This can be a probable question, classification of Vibrio. Based on salt requirement, you can classify Vibrio into non-halophilic Vibrios and halophilic Vibrios. Halophilic means, halo means salt, philic means love salt, salt loving. So those Vibrio which can tolerate higher salt concentration, say 7 to 10% salt concentration, you call them as halophilic Vibrios, and the best examples are Vibrio parahemolyticus, Vibrio alginolyticus, and Vibrio vulnificus. So these Vibrios, they can grow well in seawater because seawater contain a higher concentration of salt. So these can be associated with seawater related infections, like it can be wound infections or food infections, seafood poisoning and all. So remember the names, you don't have to learn them in detail, but the names you can by heart: parahemolyticus, alginolyticus, and vulnificus. Whereas coming to our important one, Vibrio Cholerae, it is non-halophilic. That means it doesn't require salt for growth. The other one, they require higher salt concentration. But Cholerae, Vibrio Cholerae, doesn't require higher salt concentration. Just 1% salt is optimum for them. They can grow even without salt. So they are non-halophilic. And this is the important pathogen which causes the disease cholera. So that is what we are going to learn in this session. So the most important member, Vibrio Cholerae, and what does it cause? From the name itself, it's cholerae, so it causes cholera. And morphology, again, this can also be a question. Vibrio Cholerae morphology. This is what I showed you. That is, it is having a single polar flagella. You can see the polar flagella arising from one end. So, and what is the shape of this bacteria? It is curved shaped. It is like this. It is comma shaped or very curved comma shaped gram-negative bacilli. And what is a motility called? Darting motility. Darting means actively motile with a single polar flagellum. And if you take, in patients with severe diarrhea, mucus diarrhea, you take the mucus, you stain it with gram staining and appear under microscope, you will see them like this. They will be going like this, fish in stream appearance. It is comma shaped bacteria, no? It will be moving in parallel rows. It's called fish in stream appearance. You can see that here. Parallel rows of bacteria going in a stream, going in a single parallel way. So that is called a fish in stream appearance. It looks like a number of fishes going together. That's why it's called fish in stream appearance. So regarding the antigenic structure of Vibrio Cholerae, like what we have learned for Salmonella, this one also has got a somatic O antigen, which is actually the lipopolysaccharide, and the flagellar H antigen. And based on the O antigen, we have a classification for Vibrio named after two people, that is Gardner and Venkataraman. They have done this classification. There are more than 200 serogroups based on the somatic O antigen. of which the O1 serogroup is the most important one, which has caused a number of pandemics of cholera. See, when you read history, you can see a number of pandemics of cholera has been reported, which has led to death of a number of people. And O1 serogroup is responsible for most of the pandemics. And when you take O1 serogroup, it has got two biotypes. So biotypes means it will be showing different kind of biochemical reactions. So each O1 serogroup shows two biotypes. It can be L-TOR biotype or classical biotype. Actually, the classical biotype was causing most of the pandemics, but recently they have identified the L-TOR biotype also causing cholera outbreaks. And when you take each of these, either L-TOR or classical, you have three serotypes, Ogawa, Inaba, and Hikojima. So these three are the serotypes. So O1 can be of two biotypes, L-TOR or classical, two biotypes, and each of these biotype can be of three serotypes, Ogawa, Inaba, and Hikojima. So that is how we put the classification cascade of Vibrio. There are different serogroups. It's not just O1. There are more than 200 serogroups, which O1 is more important, with two biotypes and three serotypes. So biotype of Vibrio cholera O1, classical and L-TOR Vibrios. And how you differentiate these two, the most three important tests for differentiating L-TOR from classical is L-TOR shows a positive chick RBC agglutination. If you mix the colonies with chick RBC, they will produce agglutination. If you inoculate them on sheep blood agar, if you inoculate them, they cause hemolysis of sheep RBC. And if you put a polymyxin B antibiotic disc, this one is L-TOR is sensitive to polymyxin B. So these three are the important reactions by which or test by which you differentiate L-TOR from classical Vibrio. You have another group of Vibrio called non-agglutinating Vibrios. That means O1 is the most common serogroup which causes pandemics, and there is one more thing which is causing classical cholera like picture, O139. So these two are the most common serogroups. In addition to these two, if you are getting some other Vibrio groups, anything which does not agglutinate with O1 antiserum and O139 antiserum, you call them as non-agglutinating Vibrios or non-O1 Vibrio cholerae. So O1 and O139 are the most common serogroups which causes outbreak of cholera. And if you have something other than O1 and O139, you call them as non-agglutinating Vibrios. Coming to the pathogenesis of Vibrio cholerae, so I told you O1 and O139 causes the classical cholera-like picture, which is an acute diarrheal disease. And what is the source of infection? It can either be a patient or can be a carrier. So natural infection, it occurs only in humans. There is no animal infection, and mode of infection is via contaminated water and food. So any kind of diarrheal disease, enteric disease, it will be through ingestion, ingestion of contaminated water or food. So infective dose is very high. So you can see it is 10 raised to 8 Vibrios are required to cause infection, because otherwise if the inoculum load is very less, most of them will be destroyed by the acidic pH of the stomach. So acidity of the stomach, acidic pH of the stomach will destroy most of the Vibrios. But if you have a very high inoculum, like more than 10 raised to 8 pathogenic Vibrios, it will cause infection. It will bypass the acidity. So what is the important virulence factor for Vibrio? See, Vibrio cholera, it is a toxin producing bacteria. It produces an exotoxin, which is an enterotoxin. We know anything which causes an enteric illness, the toxin will be enterotoxin, like we have learned for different kind of gastroenteritis and other things. Enterotoxin, it's an enterotoxin means it's acting in the intestine. So cholera toxin is an enterotoxin, and it's a heat labile exotoxin. See, all these are produced toxins which are produced outside from the bacteria. Those are called exotoxins. So it's protein in nature, like what you have learned for Corynebacterium bacterium diphtheria. The toxin was having 2 fragments. Similarly, this one also has got 2 fragments, but it's like this: A fragment with 5 B fragments surrounding it. So one A fragment in the center with 5 B subunits surrounding it. So what is the use of B? B is always for binding, A is always for action. So this A has also got 2 fragments, A1 and A2. A is actually split up into A1 and A2. So B is just for binding. You can see B binds to the ganglioside receptors on the enterocytes in the intestine, and thus A is internalized. When A comes inside, it goes to A, it splits into A1 and A2. A1 is having all the activity. And what does A1 do? You can see the action of A1 is it will increase adenylate cyclase activity. It increases the action of the enzyme adenylate cyclase. And what does it do? It will increase the CAMP concentration. So when CAMP concentration is increased, what it will do is it will cause outpouring of water and electrolytes, sodium, chloride, carbonate, potassium, everything is outpoured from the mucosa into the lumen, and it will reduce the absorption of sodium and chloride. So when there is outpouring of electrolytes, water also goes along with it, leading to diarrhea. So that is the reason for cholera diarrhea. That is the main virulence factor responsible for this is the cholera toxin, which is an enterotoxin. So how does Vibrios adhere to the epithelium? If this is the intestinal epithelium, the Vibrio bacteria has first got to adhere to the epithelium. Its motility is one factor which helps it in traversing this, but it has to adhere. For that, it has got a special fimbrial that is called toxin co-regulated pilus. Pilus means fimbria. So toxin co-regulated pilus is the fimbria which has got a special fimbria for attachment. Then produces the enterotoxin. What is enterotoxin? That is the cholera toxin. And how does it act? It increases the CAMP production. So what will happen? It will cause outpouring of water and electrolytes from the mucosa. So there will be increased expulsion of water, sodium, chloride, everything outside into the lumen, and there will be decreased absorption also. So all these things will lead to severe watery diarrhea. So what happens when there is severe watery diarrhea? There will be massive depletion of water and electrolytes. You know how you describe the classical stool appearance in cholera? You call it as rice water stool. So it will look like rice water. It will be because it will be having plenty of mucus flakes. So it is called rice water diarrhea. This leads to severe dehydration. That is the problem with cholera. Cholera, since it is causing massive depletion of water and electrolytes, there will be severe dehydration for the patient. So when there is severe dehydration, what happens? There will be hemoconcentration, anuria, and hypovolemic shock. BP will be very low because of dehydration. So that can lead to hypovolemic shock. So if you don't intervene, if you don't treat the patient at the right time, it can lead to shock and death. So the mortality rate, see how big is the mortality rate? 60 to 70%, and it's mostly due to renal failure. If you don't treat the patient, if you don't correct the dehydration, you have to give them plenty of fluids, intravenous fluids, and correct the dehydration at the right time. There are two types of carriers for cholera. There is convalescent carrier and chronic carrier. Convalescent carrier means a person who has recovered from cholera. For some more time, like 4 to 5 weeks, they continue to excrete the bacilli in feces. So they are called convalescence, means the recovery period. So they excrete bacilli during the recovery period. And what is meant by chronic carrier? Those are found in endemic areas where vibrios are excreted in feces intermittently from the gallbladder. See, this bacilli will survive in the gallbladder, and for a long period of time it gets excreted in the feces for a prolonged duration. Those people are called as chronic carriers. So all these people can act as source of infection. Not only a patient, carriers can also act as source of infection. Now, how we are going to diagnose cholera? Like in any infection, the first point should be you should think about the specimen. So what is a specimen here? It's a watery stool. Suppose if the patient cannot get a watery stool, you can go for an alternative specimen that is called a rectal swab. You can take a swab from the rectum, and you have to collect this prior to start of antibiotics. So don't hesitatingly, you don't have to start the antibiotics immediately. It's very important to correct the dehydration. Just put them on fluid supports and collect the sample for confirmation of diagnosis. Only then can you start the antibiotics. And when you collect the sample, it should be transported immediately to the laboratory. There should be no delay. If suppose there is delay, you have to use a transport medium or holding medium. You call that as holding medium because it will hold the bacteria as such without causing any change. So the best transport medium that we use for Vibrio Cholerae, it's named after Indian person Venkataraman Ramakrishnan medium. And see the pH of this medium. The pH of this medium is 8.6 to 8.8. So it's alkaline pH. Cholera needs alkaline pH. Vibrio Cholerae needs alkaline pH. Alternatively, other than this VR medium, you can also use Carry-Blair medium, which is used for Salmonella and Shigella, and the pH is 8.4. So if you have an alkaline pH, this bacilli will survive in it for longer periods. So you don't have to worry about this bacteria becoming unviable. So it will have a longer viability when you put it in a transport medium. So when it reaches the lab, what will you do if you get the sample in a transport medium or if you get the sample in lab? Immediately you can perform a motility testing by hanging drop method. You take a slide, you put a drop of the sample. It's a special kind of slide with a depression. You put a cover slip and you examine under the microscope. Then you can see that darting motility. This bacilli will be moving, rushing fast through the one field to the other. That's called the darting motility. So if you are able to demonstrate darting motility, you can be almost sure that this particular sample is containing Vibrio cholerae. And if you do gram staining from the mucus flakes, you might be remembering this picture which we saw in the first one. It will be like fish in stream appearance. This bacteria seen in parallel rows. So this is Venkataraman Ramakrishnan medium, Carey Blair medium. Everything will come in a screw-capped bottle to transport the sample. This bacteria is aerobic, so when you think about the culture of Vibrio cholerae, you have to be sure that this is put in an aerobic incubator. It needs oxygen and the temperature is 37 degrees Celsius and it grows best in alkaline medium. The optimum pH for growth is 8.2 alkaline pH. So like how we learned for typhoid in Salmonella, hope you might be remembering that we needed an enrichment growth and selective medium. Why? Because we are inoculating stool samples. Stool samples may contain other commensals, coliforms. You want to inhibit them. You want to stimulate the growth of only the pathogenic organism. So for Vibrio cholerae also, you require an enrichment growth and a selective medium. And I'm repeating, enrichment growth is liquid, it's broth, and selective is for solid. Selective is solids, agar. So what enrichment growth we use? We use alkaline peptone water. We know peptone water is a liquid medium, no? So if you make the peptone water alkaline by adding salt to it and making the pH 8.6, it will become a very good medium for Vibrio cholerae. So if you inoculate the stool sample into alkaline peptone water and keep it for 6 to 12 hours, what happens? Cholera will multiply, Vibrio cholerae will multiply in it and all other coliforms will get destroyed. So you have only Vibrio cholerae left in the medium. Now we are going to inoculate this alkaline peptone water, growth in alkaline peptone water onto solid media, which is the selective media. So which selective media we use for Vibrio cholerae? We use TCBS. TCBS is thiosulfate, citrate, bile salt, sucrose, agar. You have to by heart it because at least one selective medium of Vibrio cholerae we want to write for the exams when it is asked. The pH is 8.6. TCBS, and another selective medium is bile salt agar and the pH is 8.2. So TCBS and bile salt agar are the two selective media for Vibrio. Again, I am telling you, Salmonella, what was the selective medium? Selective medium is XLD and DCA. And what was the enrichment growth for Salmonella? It was called selenite F growth. So both Salmonella and Vibrio cholerae, we should remember the selective media and the enrichment media. Because it is stool sample, we require selective and enrichment media. So also you can inoculate the sample to McConkey agar, blood agar and nutrient agar are common media. We can inoculate to all the common media and you know how it will look like in all these media. In nutrient agar, no, if you put it in nutrient agar, you will see disc-like, round disc-like colonies with slight bluish tinge. And in blood agar, they will have colonies with hemodigestion. It is not hemolysis, but hemodigestion showing some kind of greenish discoloration because Vibrio cholerae can cause hemodigestion. And if it is an L-Tor biotype, it can produce hemolysis in sheep blood agar. And when you put it in McConkey agar, like Salmonella and Shigella, you can see here McConkey agar, Vibrio is also non-lactose fermenting colonies. So it is pale color colonies. And how it will look like in TCBS? TCBS is a green color medium because it has a green color indicator. When Vibrio grows in it, it will ferment sucrose in the medium, changing the medium pH. So it will produce yellow color colonies. The yellow color colonies in TCBS is because of sucrose fermentation. And when you do gram staining from the colony, what will be the appearance of the gram staining? It is a comma-shaped bacilli, gram-negative comma-shaped bacilli. If you do hanging drop preparation again from the colony, you can see the darting motility. Thus, we can confirm it as Vibrio cholerae. Now, further confirmation is done by the biochemical reactions. I told you it ferments a number of sugars, including sucrose, but it doesn't ferment lactose. That is why McConkey agar shows non-lactose fermenting colonies. Vibrio will produce acid, but gas is absent. No gas production for Vibrio. It is catalase positive, and this reaction you should not forget Vibrio Cholerae, it is oxidase positive, it is indole positive. We can reduce nitrates to nitrates. There are certain other specific tests for Vibrio like a string test positive, nitroso-indole positive, of which the better one you have to remember it's sucrose fermenting, non-lactose fermenting, catalase and oxidase positive, indole positive. How we are going to treat? So now we know the patient sample contained Vibrio Cholerae. We have come to the diagnosis. Now we want to treat the patient. So we are going to treat the patient by providing plenty of fluids. You want to correct the dehydration. So we are going to give fluids, plenty of fluids. Suppose the patient is having severe vomiting, they won't tolerate the oral fluids. Then you have to give IV fluids. So intravenous fluids should be given, and you can also give antibiotics, of which tetracycline group of antibiotics is active against Vibrio group. So you can give doxycycline, and you can give some supplements like zinc supplements, which is good for controlling diarrhea. So prompt water and electrolyte replacement is very important. So you have to give appropriate IV fluids, and a better one is a ringer lactate solution. And oral rehydration therapy can be given if the patient is withstanding oral fluids, if there is no vomiting. And the best antibiotic of choice for Vibrio Cholerae is doxycycline. But more than antibiotic, the best part, the most important part of treatment is correction of dehydration or rehydration therapy. Coming to prophylaxis, you have general measures and specific measures. If it is a foodborne, waterborne disease, always you have to think about the general measures. So it is like improving the sanitation, purification of water samples, better sewage disposal. And if a patient is found to be infected, they should be isolated and the excreta should be disinfected properly, and contacts and carriers should be followed up. Now, specific measures, we have got some vaccines for Vibrio Cholerae. A killed parenteral vaccine is available. Killed oral vaccine is available. A live oral vaccine is available. Suppose if you want to give a chemoprophylaxis, that means if you suspect that a particular person has been exposed to a Vibrio Cholerae, if there is an outbreak in an area, all those people who have consumed that particular water or food. Similarly, during floods, we know there is high chance of spread of cholera and leptospirosis. We might have heard about the prophylaxis with doxycycline then. So doxycycline is not only used for treatment but also for prophylaxis. So the important questions from this part, from Vibrio, you can get about the morphology. Remember the comma-shaped bacilli, the pathogenesis, toxin. Don't forget the cholera toxin. Here, don't forget the comma-shaped gram-negative bacilli and the darting motility with a single polar flagellum. And lab diagnosis, remember all the culture media we use for this enrichment and selective, and treatment and prophylaxis. So anything can be asked. It can be a probable essay question or short note question. So please be thorough with all these points. Thank you.